No.
Okay.
Now we can, yes. Perfect.
Okay. Welcome, everyone. Good morning. I'm going to present this research, which is titled Evaluation of Antimicrobial and Enzymatic Activities of Lactic Acid Bacteria Isolated from Fresh Vegetable Sources in Chihuahua, Mexico. Okay, that was the end of the presentation. I'm sorry. Okay, here we go. Lactic acid bacteria have gained importance in the last years since they are an almost inexhaustible source of metabolites. The discovery of novel strains or improved metabolites that might have technological applications has become a challenge for the food industry. For example, lactic acid bacteria with antimicrobial activity is deemed to be a harmless approach to maintain safe food processing. They can ferment and protect, becoming a sustainable solution for food protection and avoiding the use of chemical products. You can see in this table several antimicrobial activity evaluations on lactic acid bacteria. Those lactic acid bacteria were isolated from vegetable sources and had antimicrobial activity against several pathogens like molds, gram-positive and gram-negative bacteria. Another potential application has enzyme sources, since they have been reported that microbial enzymes are more desirable because they have presented enhanced catalytic activities and can be easily produced in economic culture media. The primary enzymes employed for food making are lipase, protease, and amylase. And citrate metabolism is a key feature for lactic acid bacteria in the dairy industry. These are several enzymatic assessments on lactic acid bacteria, where you can see protease, lipase, citrate metabolism, and amylase. Thus, this research was directed to assess the antimicrobial activity of lactic acid bacteria by microtitration method and the enzymatic activity, protease, lipase, amylase, and citrate metabolism. The fruit samples were placed in a plastic bag with a maximum recovery diluent, and an electrode was transferred to tubes with MRS broth. Tubes were incubated, and positive tubes with growth were strict on MRS agar, were incubated again, and the typical colonies, gram-positive and catalase negative, were strict again to obtain a pure culture. For the antimicrobial activity, brain heart infusion broth supplemented with dipotassium phosphate to avoid the action, the effect of the lactic acid, suspension of indicator microorganisms, and lactic acid bacteria supernatant were placed in a microplate, and the optical density was measured before incubation, before and after incubation. The microplates were incubated with the following incubation conditions according to the indicator microorganisms. Those microorganisms were gram-positive, gram-negative bacteria, lactic acid bacteria, molds, and yeast. For the enzymatic activity test, 4 media were prepared: milk agar for protease, tributylene agar for lipase, citrate agar for the citrate metabolism, and starch agar for the amylase activity. And the data were analyzed by a hierarchical cluster analysis employing a Minitab 18 software with an 85% of similarity. For the molecular identification of the lactic acid bacteria, the DNA was extracted according to the reported by Postgich and Newman, and the DNA extracted was amplified. The 16S rDNA gene was amplified using these primers 27 forward and 1492 reverse. And the amplified samples were sent to Microgene Corporation to be sequenced. And we can see in this table the vegetable sources for lactic acid bacteria. These samples, the availability of samples depended on the season, and we wanted to find this, vegetable samples without treatment. That was another issue sometimes. 76 lactic acid bacteria were isolated. All of them were gram-positive, catalase negative, varied from coccus, bacillus, or coccoids, grouped in pairs, tetrads, chains, and cluster. And the supernatant, the pH of the supernatants varied from 3.9 to 4.20. This figure is a dendrogram that was built by the hierarchical cluster analysis. You can see 13 clusters, and the lactic acid bacteria supernatants were gathered in 13 clusters, but cluster 1 is the cluster that grouped the lactic acid bacteria supernatant with the highest antimicrobial activity against the indicator microorganisms. These lactic acid bacteria are presented here. You can see the percentage of inhibition. For example, Listeria monocytogenes was very inhibited. Also, Lactococcus lactis, Saccharomyces cerevisiae, Candida albicans, Candida tropicalis. And regarding the enzymatic activity, 60% of the samples were protease positive and 80% were lipase positive. Almost half of the samples were able to metabolize citrate, and only 6.6 were able to hydrolyze starch. These are examples of the antimicrobial activity test, where you can see the clear halo around the growth, which represents the hydrolysis of casein. And this intense blue color is the production of Prussian blue. That means the lactic acid bacteria uptake the citrate in the media. Another example, you can see the clear halos around the growth, which are the hydrolysis of tributylene acid, and the clear halo when the plate was flooded with iodine, which means the absence of starch. And this dendrogram was also built by the hierarchical clustering analysis. The lactic acid bacteria strain were gathered according to the same characteristics. For example, cluster 1 gathered protease positive strains, and cluster 2 mostly citrate metabolism positive strains, and cluster 5 amylase and citrate citrate metabolism positive, but we can highlight 8 strains. You can see them here. They were selected for molecular identification because they presented at least 3 enzymatic activities. Only one strain, which was isolated from tangerine, showed the 4 enzymatic activities. And the strains selected by their enzymatic and antimicrobial activity were identified. You can see 5 of them were Enterococcus munti, 5 more were Leuconostoc mesenteroides, and a couple of them were Enterococcus faecium. Okay.
Thank you very much, Rafael. Jose, sorry. So what could be the conclusion of your work, Jose?
Give me a second. Okay. The conclusion, well, lactic acid bacteria isolated from vegetable sources. As you can see, they have important characteristics. For example, those strains that had that presented 3 enzymatic activities, some of them were also had antimicrobial activity. And for example, those strains with protease and lipase and citrate metabolism are useful for the dairy industry. And also, those strains with amylase activity are useful for the pickling process, and were aid with antimicrobial activity. They can, as I said at the beginning, they can ferment and protect. And also, they have those enzymatic activities that are important.
Okay, Jose, I think there's a very big potential in following this work and working with these microorganisms, as they have very good enzymatic activities. And as you said, some of them have more than 2 enzymatic activities, right? So
Yeah.
I congratulate you, very interesting work. And we can move now to our next presentation.